Okay, I'll admit it. I've done it. I've occasionally worn makeup past the expiration date. 3 years expired, 2 years expired. I don't even know how old this is. This has lasted me longer than some relationships, just saying. And even though beauty experts tell us to toss our expired cosmetics out, you know what? They're expensive. And I'm not always done with what's in the bottle by the time the expiration date hits. Besides, when I apply it, everything seems fine, right? Now when I see mold in my makeup or it smells funky, obviously I throw it out. But what about the stuff I can't see? Like bacteria.
They're really, really small, microscopic.
So it's possible I can't even see what my makeup's contaminated with. But why sit here and wonder when I can use science to find out? So my science goals are going to be to find out if there's anything nasty lurking in my expired cosmetics.
All right, let's gear up.
I'm here at Counterculture Labs in Oakland, California, where citizen scientists can get their geek on. This is molecular biologist Dr. Chris Noel.
And from my pile of expired cosmetics, we selected a few to test.
I have a lipstick, 2 years expired. I have a lip gloss, 4 years expired. My mascara, 8 months expired. And my concealer, only 1 month expired.
First, we're going to dilute the cosmetic and spread it, along with any bacteria, onto an agar plate.
Looks like Jell-O.
That's not that far off. Agar is derived from algae and behaves a lot like the powdered stuff you buy in the grocery store.
But cosmetics have preservatives. Shouldn't that prevent bacteria from growing?
Yes, but preservatives become less effective over time.
So even though not required by law, this could be why some cosmetic companies still offer expiration dates. How do we know if there's any bacteria even in here?
Well, we have to make sure we grow enough to seed, but that can take time.
What can I do to encourage them to grow quickly?
Well, we feed them. Bacteria can't eat the agar, but they can absorb the moisture, the salt, and the nutrients such as carbon and nitrogen sources from it. We also keep them warm to encourage them to grow. This way to the incubator.
Bacteria reproduce through a form of asexual reproduction called binary fission. The bacteria duplicate their genetic material and divide, producing 2 new identical copies. It's a solo endeavor. One cell becomes 2, then 4, then 8, and before you know it, one bacterium has produced millions of cells in just a few hours. Geez, bacteria, get a room.
With yourself.
Oh, hey, this is pretty cool. These millions of cells form a little pile called a colony. Each colony has only one species of bacteria, and there are many colonies on a plate.
And now that they've grown up, we can use these properties such as size, shape, color to identify them. Look how different the colony morphologies are. One is wrinkly, dark blue and tight. The other is like a fuzzy cloud-like spread out colony.
With some bacteria though, their identity can be a little more ambiguous. Fortunately, we have DNA sequencing to isolate a portion of the DNA and magnify it, so we can have better species identification. We got some really cool results, didn't we? Thank you so much for your time.
My pleasure.
Now that we've completed our lab work, we're gonna head over to the California Academy of Sciences. I'm here to speak with Dr. Shannon Bennett. Not only is she the Chief of Science and the Dean of Research, she's done a lot of work on infectious diseases and can help us interpret the results. I tested my expired cosmetics and got a lot of interesting results. Can you help me understand what they all mean?
Sure, I'll try.
In my lipstick, I found Staphylococcus epidermidis and Staph werneri. Am I gonna get a staph infection?
So Staphylococcus is a genus of bacterium that's definitely associated with infection. And the most famous of that party is Staphylococcus aureus, and it's serious. But it turns out that Staph aureus, Staph epidermidis, staph warneri, they're parts of our normal skin community if there's a situation in which your immune defenses are compromised in some way and they have the opportunity to get introduced into the inside of your body, they can cause infection.
In my lip gloss, I found Bacillus cereus. Is that a serious problem?
If you had chapped lips or you had a crack in your lip and they maybe got introduced into your tissues from outside to inside, they actually do have the ability to release toxins that can break down your tissues.
In my mascara, I found Panawea agglomerans. Am I saying that right?
Panawea agglomerans. We call that a fecal indicator because sometimes it's actually used to study when a certain part of the environment has been contaminated with poo. So I would actually go so far as to say that your mascara has become contaminated from a fecal source.
In my concealer, I found Bacillus megaterium. Is that a big deal?
So Bacillus megaterium really is harmless. It has not been associated with any infections that we know about, so we're pretty sure it's one of the good guys, a harmless bacterium. In fact, it might even go so far as to keep other harmful bacteria in check. We have these complex communities of bacteria with many species, and they're outcompeting each other, fighting each other for resources. And then as time goes on, that community can become skewed, and bad guys, for example, can become over-dominant.
I asked Dr. Bennett if she wanted to share any final thoughts.
When a manufacturer stamps your makeup with an expiration date, the reason that makes some kind of sense is because over time, the balance in that microbial community can become skewed. And what we don't want is for certain kinds that can cause disease to become over-dominant or super numerous, and then they're really able to cause a problem. So I have a great respect for expiration dates for that reason.
Dr. Bennett, thank you so much for your time.
You're very welcome.
Does this video change your mind about how long you keep your makeup? Were there any surprises, or do you have any thoughts you want to share? Put them in the comment section below. Lastly, I'd like to thank Google's Making in Science team for providing the resources to make this video possible and allowing YouTubers like me to share how we're using science to explore the world around us.